membrane, basolateral membrane. Tell me what you are watching here. Which transporter?
We see from the apical side, we see first the sodium hydrogen co-transporter, exchanger.
Exchanger.
And we see the sodium, 2 calcium, 1 potassium co-transporter. So it takes in, 1 sodium, 1 potassium, and 2 calcium ions.
Chloride.
Chloride, sorry.
Potassium 2 chloride co-transporter in apical membrane. Just you have to draw it. Okay? I think twice or thrice you can keep it in your mind. Got it? And what do you see here?
We see potassium, sodium, ATPase.
Sodium, potassium pump or sodium, potassium ATPase.
Chloride channel.
Channel. It means it is channel, leak channel. Do you know channels?
Yes.
It is leak channel for chloride ions.
And here we see potassium also leaks out from both sides.
Potassium leaks here into the lumen and it is reabsorbed in this direction. Okay. When potassium comes in the lumen, what happens regarding the charge?
The charge in the lumen becomes more positive.
More positive. Okay. And what happens?
This causes calcium and magnesium to leak through the paracellular path into the other side. Do we know
which path?
Paracellular.
Paracellular pathway. Got it? No question. And loop diuretics such as furosemide inhibits sodium potassium 2 chloride co-transporter. When you see our factors are moving together, say co-transporter. In the opposite direction, we say exchanger or counter-transporter. Got it? Okay. Here you can see a cell in early distal tubule. What do you see here?
We see sodium chloride transporter.
Okay, co-transporter or transporter in the
Apical surface.
And in here?
We see chloride, we see sodium, potassium, ATPase, and chloride channel.
Okay, and
Chloride
channel. Thiazide, actually diuretics inhibits these transporter. What happens after that? Sodium cannot be reabsorbed. It remains inside the urine, and actually the body gets rid of excess sodium. Okay, it is administered in people with hypertension. Got it? No question.
No.
Okay, now we are going to talk about late distal tubules and cortical collecting duct and cortical collecting tubules. Here in the apical membrane, we have
sodium and potassium channels.
here in late distal tubule and cortical collecting tubule, cortical collecting duct. Ducts are larger than tubules. Okay. We have two types of cells. Okay, principal cells, intercalated cells. Okay. Here you can see one principal cell in the apical membrane. Sodium leak channels for sodium, leak channel for potassium. When we have leak channel, it means ions are moving based on electrochemical gradients. Okay? Sodium concentration is low, sodium comes inside, and potassium goes outside. Actually, this driving force is provided by existence of sodium potassium pump. Okay, here, don't forget. As I told you, you cannot see sodium potassium pump in the apical membrane. Got it? No question? And Sodium channel, we have sodium channel blocker, which block this channel, and aldosterone antagonist. At here, we will talk about that. No question?
No.
Here you can see interconnected cells. Interconnected cells are two types, A and B. These cells are very important or in acid-base regulation. Okay. Here you can see type A cell. Carbon dioxide plus water actually forms carbonic acid. Carbonic acid forms bicarbonate plus hydrogen. Hydrogen is secreted by hydrogen ATPase and hydrogen potassium ATPase. Bicarbonate is reabsorbed by bicarbonate chloride exchanger in the opposite direction. Okay, it is active in acidosis or alkalosis.
Acidosis.
Acidosis, why? Because we have excess hydrogen that
Excrete.
must be excreted. But you have to actually keep in your mind the location of transporters and channels, and after that for interconnected type B, you have to change the location. Here again, we have hydrogen and bicarbonate. Bicarbonate is secreted by bicarbonate chloride exchanger. On the other side, hydrogen is reabsorbed. It is active in alkalosis when there is excess bicarbonate in our body. But the channels are completely the same. Okay, just you have to memorize it. Regarding medullary collecting duct, actually two main factors are reabsorbed from medullary collecting duct. One is urea. Do you remember yesterday I told you we have 50% of urea is reabsorbed. One is urea reabsorption and the second is water reabsorption. Both in the existence of ADH. When we have ADH, urea and water together begins to actually reabsorb. Got it? Medullary collecting duct. Okay, we will talk about in the next part. What is ADH, urea transporter, and also water reabsorption. Now we are going to talk about regulation of reabsorption. Yesterday I talked about regulation of GFR. Today we are going to talk about regulation of reabsorption. Do you remember the first item was in GFR regulation? First item was sympathetic. Written in this way. Sympathetic activation. Do you remember?
Yes.
I told you mild and moderate stimulation has no effect on GFR. Okay, the story is the same regarding reabsorption. Mild or moderate stimulation has no effect on reabsorption. Okay, reabsorption. But no effect on reabsorption. But severe stimulation, actually it is considered to be an alarm for our body. Okay, it means, for example, there is hemorrhage or there is brain ischemia. As I told you yesterday, here GFR decreases, reabsorption increases. GFR decreases, reabsorption rate increases. Why? I told you arterioles begin to constrict a lot by severe stimulation. Okay? Actually, it increases PG decreases, it increases, GFR decreases, okay, and at the same time, it decreases the pressure in efferent arteriole more than normal. For example, here, the pressure is, for example, instead of 30 millimeter of mercury, it is, for example, 10 millimeter of mercury. It was an opposing factor for reabsorption. Pressure here in the pretubular capillary decreases, it helps reabsorption. Got it? Was it clear? Okay. And another item was some hormones. Okay. 1, 2, 2. Hormones. Which hormones are important?
Vasoconstrictors and vasodilating hormones.
Aldosterone,
Yes.
ADH,
Show me.
and GNO.
These hormones are very important. First is aldosterone. It is very important. The site of action. You have to memorize the site of action. It has effect on collecting tubule and collecting duct. And what's the effect? The effect is reabsorption of NaCl, reabsorption of water, secretion of potassium, and secretion of hydrogen. Okay, when potassium concentration outside the cell increases, aldosterone is released, is secreted. When we have blood pressure decreases, when blood volume decreases, okay, aldosterone is released, and actually the site of action of aldosterone is on principal cells. Do you remember principal cells in late distal tubule and cortical collecting duct and tubules? Okay, it increases the activity of sodium potassium pump, and in turn it increases sodium reabsorption and potassium secretion. Got it? Another one is angiotensin 2. In all portions of tubule, you can see it has, actually it can act, and the effect is increasing NaCl and water reabsorption, increasing hydrogen secretion. Okay, here you can see actually the action of angiotensin 2, effect on the receptor is AT1. Okay, and after that, after activation of receptor, sodium potassium pump increases the activity. It increases the activity of sodium bicarbonate co-transporter. It increases the activity of sodium hydrogen exchanger. Okay, actually, angiotensin 2 stimulates aldosterone secretion, constricts the efferent arteriole, as we talked about yesterday, reduces pretubular capillary hydrostatic pressure, raises filtration fraction. We have talked about these items yesterday. Do you remember? No? Or yes? Directly stimulates sodium reabsorption. No question? And the next one is ADH or AVP. ADH is the same as AVP. Arginine vasopressin. Okay. As you can see here, receptor is V2. After binding, when there is water deficiency, AVP or ADH is released. After binding to receptor, GS is stimulated. Adenine cyclase is activates in the membrane. cyclic AMP is produced as a second messenger. After that, inside the cell, in which part? In the late part of nephron, but in the last part of nephron. From late distal tubule on. Okay. Inside the cell, we have some vesicles. In the membrane of these vesicles, you can see aquaporin 2. These vesicles contain aquaporin 2. Okay. After binding AVP to the receptor, these vesicles begin to join the membrane. Now we have aquaporin 2 in the membrane and water can be reabsorbed. Got it?
Yes.
And don't forget, aquaporin 2 in the apical membrane is sensitive to AVP or ADH. Okay. But here you can see in the basolateral membrane, you can see aquaporin 3, aquaporin 4. All the time they are here. They are present here. Okay, they are not sensitive to ADH or AVP. Got it? If I ask you regarding which aquaporin, for example, response ADH, you have to answer aquaporin 2. Okay. The next one is ANP. Do you know ANP? Antriol, natriuretic peptide, ANP. Suppose actually ANP is released from atrial when blood volume increases. Okay, there is the cells in the atrial begins to stretch. Okay, and this stretch causes these cells to release ANP. And what's the effect of ANP? Actually, ANP has effect on distal tubule, collecting tubule, and collecting duct. Okay, it decreases sodium chloride reabsorption. And the last one is thyroid hormone. It's not important in renal physiology. Actually, in endocrine physiology, you have studied these hormones. No question? Regarding clearance, what's the definition of clearance? Volume of plasma that is completely clear of a substance. What does it mean? It means suppose that here we have some sodium. Okay, again here is glomerular capillary and Bowman's capsule. Okay, we have some sodium. Okay, the clearance means volume of plasma which is completely cleared from sodium. Is it possible?
No.
No. Why? Because I told you based on simple diffusion, sodium begins to move. And the concentration is the same. It means it's an imaginary state. Okay, just by this definition, we are gonna talk about to quantify kidney function. Got it? For clearance, calculation. Suppose that we are gonna calculate clearance. For calculation, we use from a substance named inulin. As I told you yesterday, inulin is not
Made by the body.
is not produced in our body. Actually, it is from the roots of some plants. Okay, for calculation of clearance, sorry. Actually, a person must be hospitalized because this substance must be infused intravenously. Okay, it is very difficult. What's the properties of inulin? Inulin is virally filtered. What does it mean? When you say something is virally filtered,
Filtered
it
like
means
water.
the concentration of that substance is the same in glomerular capillary and Bowman's capsule. Okay, the first properties. The substance must be virally filtered. It means we cannot use from fatty acid, calcium, and proteins for calculation of clearance. Why?
It's not really the same amount.
Because there is not the same concentration. Thank you. Because I told you half of calcium and most fatty acid bound to plasma proteins, and they cannot be freely filtered. Got it? And the second property is no reabsorption. Actually, inulin not be reabsorbed in the tubule. Got it? And the third property is the substance must actually we have to have no secretion. No secretion. Inulin is not secreted into the tubules. If we have a substance with these properties, we can use for clearance actually estimation. Okay. And what's the formula? Here we say the amount of filtered amount is equal to excreted amount. Am I right? Amount filtered is equal amount excreted. Do you agree with me? Yesterday I told you you can calculate amount of substance which is filtered by this formula. GFR multiplied by P plasma concentration of substance S. Do you remember?
Yes.
And equals excreted. Amount excreted actually is calculated by this formula. US concentration of substance in the urine multiplied by V urine volume. Okay? Urine volume. This one is concentration of S substance in the urine. Okay? Concentration of substance in plasma. And this is G. Okay? I want to calculate GFR. GFR equals US multiplied by V divided by PS. Got it? Was it clear up to you? You have to keep it in your mind. You have to memorize it. Okay. On the other hand, we say clearance formula, clearance of S, for example, substance S, is US multiplied by V divided by PS. It's all the same. It means when we calculate clearance S, clearance of one substance, with these characteristics, okay, it's the same as G. Got it? For inulin, clearance S is completely the same as GFR. Got it? No question? But a big problem with us is that the person must be hospitalized, and we have to have constant infusion of inulin. And it is very difficult. In these people, we use from clearance of creatinine in order to estimate GFR. What's the problem of creatinine? Actually, creatinine is really filtered, okay? There is no absorption for creatinine. But the problem with creatinine is that we have a little secretion for creatinine. It causes GFR to be estimated more than normal. Okay, but we can ignore it. But in patients, okay, in this patient, we use from creatinine, sorry, clearance in order to estimate GFR. Was it clear? Okay, now I want you to calculate. Okay, tell me please, clearance rate for glucose is 0, for a substance is 0. What does it mean?
It's entirely reabsorbed. It's not filtered.
One possibility is that it is completely reabsorbed. And the second one,
There's none of it available. There's no glucose to actually clear it.
there is no filtration. Thank you so much. There is no filtration. The substance is not filtered, the first possibility, and the second one is that it is completely reabsorbed. Got it? Don't forget, okay? I might ask you about it. Regarding sodium, for example, clearance rate is 0.9, chloride 1.3, potassium, nothing to memorize it. For urine, it is 125 milliliter per minute. Got it? Because it has 3 properties that we talked about. For creatinine, clearance rate is 140. And what's the cause of that?
Because we are
Because it is secreted, a little amount of creatinine is secreted. When a number, okay, for example, we say clearance of substance S is blah blah, okay. When the number was below GFR, below 125 milliliter, it means it is reabsorbed. There is reabsorption for that. The number above this amount means there is secretion. Got it? But 0 means there is completely reabsorption or
no filtration.
no filtration. Was it clear? Answer the question, please.
Is urine flow the same as urine volume?
Tell me the correct answer.
20?
What is the clearance of the substance with its concentration in the plasma? It means PS is 10 milligram per deciliter. Okay. Actually, you have seen in a way that you all guessed. Write it, please. You are not guessed here.
It's 20
I think C.
And the other students?
It's kind of-
Yeah.
Also thinks.
PS is 10 milligram per deciliter. Okay, deciliter equals
10 milliliters. No, 10.
100
100.
milliliters. 100. But here, now we need to change the unit. Okay, I will talk about that. PS is 10 milligram per deciliter. Concentration in urine. Concentration in urine is US. US is 100 milligram per deciliter. Urine flow is 2
Two.
milliliter per minute. V is 2 milliliter per minute.
The answer is
Put it
C.
in the formula.
C.
Do you agree with C?
Yeah.
Okay. C is correct. Got it. are
There's next reabsorption, right?
Then there's
in
secretion.
western, the other in
Then there's secretion, right?
B.
It should be D.
B.
D.
B or D.
D. D.
B.
B.
P.
No, I think D.
Freely filtered, like there's secretion or there's reabsorption.
Clearance of a substance which is freely filtered, okay, is less than that of in urine. It means there is net reabsorption. If we have a net secretion,
It's more.
the value must be more than in urine clearance. Got it? Okay.
D. F. D.
Okay, do you remember yesterday I told you the effect of blood pressure? Do you remember yesterday I told you the effect of blood pressure changing, okay, changing blood pressure on GFR and RGF is in minimum level because of two main mechanisms. Do you remember? First one was, do you remember?
TGF and T and GB.
We talked about TGF, a feedback from tubules toward glomerulus.
Gloomer.
The second one was
GT, GB, balance.
glomerular, tubular balance. It means there is a balance between filtered load. There is a balance between a balance. It means a balance, okay, not equality between filtered load and reabsorption rate. What does it mean? It means suppose that GFR is normal, 125 milliliter per minute. Okay. We say 65% of water and solute are reabsorbed in proximal tubule. Do you remember?
Yes.
Water and solute together. 25% of just solute are reabsorbed in thick ascending limb. This part is not permeable to
To
water,
water.
even in the presence of
ADH.
ADH. Do you remember? Yes? GTD says that when GFR is more than normal, what happens? Again, 65% of these GFR is reabsorbed in proximal tubule. 25% of
same?
remain constant. It's an intrinsic property in the tubules. Got it? And what's the importance? What's the importance? We say it prevents distal tubules or distal parts of nephron from overloading. Okay, we don't want to have overloading in late parts of nephron. Why? Because as I told you, actually the last portion of nephron is doing the final processing. It is very important. We have to prevent overloading distal tubule by this mechanism. Ready? Was it clear? Here you can see. Here you can see TGF. I will talk about that later. And here GTP. Okay, what does it say? It says when GFR increases or decreases. Okay, when GFR increases, the percentage of reabsorption in proximal tubule and thick ascending limb is the same. It's constant, not is the same. It's constant. Okay, it prevents overloading of fluid in distal portion of nephron. Got it?
Doctor, could you please repeat the GFR? GFR.
GFR what?
Repeat this mechanism. If it's like the
Mechanism, I don't know, we don't know by which mechanism, but suppose that. For example, suppose that GFR is 10 milliliter, okay? 65 milliliter is reabsorbed in proximal tubule, okay? And suppose that it is normal. Okay, I'm gonna take an example. Okay. Let me write over there. What does it mean? Yesterday the light was more than today.
yes, because we
I think you cannot see the board well.
We turned it off so that we could see the projected button.
Suppose that GFR, just I'm gonna take an example. Okay, normal GFR is 125. But for the simplicity, I say GFR is 100 milliliter per minute. Okay. From this amount, 65%, it means here 65 milliliter, is reabsorbed in proximal tubule. 25%, that here is 25 milliliter per minute, is reabsorbed in thick ascending limb. How much is delivered to the last parts of nephron?
10%?
10%?
Yes.
10 milliliter. 10 milliliter per minute is delivered to the late parts of nephron. Now suppose that GFR is, for example, 200 milliliter per minute. 65% of this amount is reabsorbed in proximal tubule. 65% here is 130 milliliter per minute in proximal tubule. Got it?
Okay.
25% is 50 milliliter per minute is reabsorbed in thick ascending limb. How much is delivered to the late portion?
20.
That's
20. mechanism, which is an intrinsic mechanism. Okay, and it is unknown. This GTB prevents distal tubular from overloading in order to do a final processing on urine precisely. Got it? Was it clear?
Yes.
Was it clear for you? It was your question.
Yes.
It is GTB. Here in this figure, you can compare TGF with GTB. Okay? No question? Was it clear? Do you remember net filtration pressure in GFR?
Yes.
Alhamdulillah. Okay. GFR is
125.
GFR equals KF multiplied by net filtration pressure. Do you remember?
Yes.
Yes. Reabsorption rate is again KF multiplied by net reabsorption pressure. Okay? Again here, KF depends on permeability of capillaries. Which capillaries?
Glomerular.
Here is glomerular in reabsorption. We don't deal with glomerular. Peritubular capillary. Okay, here is the properties of glomerular capillary. Here we deal with peritubular capillary. Okay, don't forget. It depends on permeability and surface area of peritubular capillary. Okay? Net reabsorption pressure. For reabsorption, I told you in reabsorption, the direction of reabsorption is in this site. Okay? It might be reabsorbed a substance from transcellular or paracellular. On the whole, finally, we have a lot of substance which are reabsorbed and are accumulated here. Where is here? Here is interstitial fluid. Interstitial fluid. Up to here, we have talked about different mechanisms, different proteins which are found in apical and basolateral membranes of different parts of nephron. Am I right? Now we are going to talk about net reabsorption pressure. Pressures which are important in determination of reabsorption rate. Okay, here you can see pre-tubular capillary. Okay. Substances are accumulated here. Again, we have P, hydrostatic pressure in pretubular capillary. Do you remember yesterday, hydrostatic pressure in
Yes.
glomerular, we said we showed in
CG.
by PG. But here we show by PC. C means pretubular capillary. But if PC is, how much is PC?
PC
13.
13.
13. Do you remember I told you efferent arteriole reduces the pressure. It is 13 millimeter of mercury. Again we have pi C. Pi C is colloid osmotic pressure in
40.
peritubular capillary, and the amount is
32.
the same as pi G.
Okay.
32. Yes. 32. Now let's talk about the pressure in IF, interstitial fluid. Hydrostatic pressure in IF, PIF, is 6 millimeter of mercury. And pi C, we have a lot of protein here. Pi IF is 15 millimeter of mercury. Tell me these are favorite or opposite? To reabsorption. Reabsorption direction is
This one.
Inside.
Pi IF is opposing.
PC
Opposing.
is opposite, but because the direction is in this side. Correct?
This.
Pi C
Is favoring.
Favoring?
is favoring because proteins are here and pull
Water.
substances from IF toward pre-tubular. Okay.
We have to.
Opposing.
IF is
Opposing.
opposing.
No, favor.
By
Oh, yeah,
this
favoring.
amount, the substance exert the pressure, but it is favoring. And pi IF is
Opposing.
opposing. Do you agree? Are the students agree with me? It is opposing. Now you have to calculate net reabsorption pressure. How much is net reabsorption pressure?
Supposed to be
28 minus 28.
13.
And the other student?
Yeah, it's 10. Net reabsorption rate is
Today
10.
some students are my guests. Welcome.
The net reabsorption rate or net reabsorption pressure is 10
Net
millimeters.
reabsorption pressure.
Is 10 millimeters of mercury.
Do you agree?
Yeah.
10 is correct. Okay, you have to sum 6. Wait.
32.
This one,
38.
38 is favoring forces. And in this direction is opposing. 13 plus 28 is opposing a lot. And on the whole, net reabsorption pressure will be 10 millimeter of mercury. But again, yesterday I told you GFR is 125 milliliter per minute. Reabsorption rate is 124 milliliter per minute. Okay, now let's calculate KF. KF equals
124.
124 milliliter per minute
On
divided
reabsorption.
by
Reabsorption.
10 millimeter of mercury. KF will be
12.4 milliliter per minute per millimeter of mercury.
how much was the KF in
12.5.
kilometer?
12.5.
It was 12.5. It means the characteristic of these capillary glomerular
It's all
capillary
point-
is different
all point.
with compared to properties of pretubular capillary. Okay, because KF is not the same. And what does it mean? It means
It means that
for each
millimeter of mercury
pressure difference between
the
interstitial
fluid
and the
peritubular
capillary.
pretubular capillary in each
minute
minute.
is
You have to
12.4.
define just based on the formula. In each minute
12.4 milliliters of fluid flows is reabsorbed.
is reabsorbed. But I want another student to talk about the definition of KF. Who is volunteer? Based on the unit. You have to define it based on the unit. Nobody is volunteer?
Other than you.
You are smart.
That is very smart.
Rafa,
No, thank you.
talk about that. Define
It would be the difference, the pressure difference
it.
between the interstitial fluid and the capillary bed of the
capillary
per minute
per minute in each
It would be 12.4 millimeters
Mercury.
of reabsorption.
Reabsorption. Got it?
No.
Okay, thank you, John.
Thank you very much.
Okay, again. If Kf increases reabsorption,
Increasing.
if Kf decreases reabsorption, decreases such as, for example, some diseases, as I told you yesterday, diabetes, hypertension, and so on. Regarding PC, actually just PC and pi C are variable. And so others are not the same. Okay, for this reason, we will not talk about that.
Doctor,
When PC,
could you please increase the size of the
yes,
slide?
of course. Yes, you're right. Sorry.
Actually, because my
When PC increases, PC increases, the blood pressure increases.
Really?
Actually, it decreases
Reabsorption.
Do you agree with it?
Yeah.
Yes.
PC might be changed as a result of actually the same as
Efferent resistance? or-
GFR, the same as GFR. When arterial pressure increases, PC,
Increasing.
it's increased.
I told you by autoregulation, this effect will be at minimal size, at minimal level. Okay? You remember sympathetic stimulation and the severe stimulation as effect on arterial pressure. Here, when resistance of afferent arterial decreases, resistance decreases means
More blood flow.
they have vasodilation. And what happens? PC
Increases.
when resistance of efferent increases. Again, PC
Increases.
it increases the inflow to pre-tubular body. Regarding pi C, suppose that pi C is increased. What happens with absorption?
Increases.
Pi C actually is changed as a result of changing in pi A. It means colloid osmotic pressure of
Efferent?
systemic circulation. Okay? When pi A increases, pi C increases. Filtration, when filtration fraction increases,
Insecure.
filtration fraction increases. Do you remember I told
Yes.
you a lot of plasma is filtered. What happens? Proteins remain in the glomerular capillary. They begin to move to peritubular capillary. Concentration is great for this reason. So it facilitates reabsorption. It increases reabsorption rate. Got it? Okay, thank you. Thank you, and I think we have finished our sessions.